bacterial infection and immunity. And we know that our subject is medical microbiology. And for a long time, medical microbiology mainly focuses on infection. So different types of infections, including bacterial infection, virus infection, and even like a parasite infection. So we are learning about pathogens. On the other hand, we learn something about immunity. So how the immune system respond to infections. And because we learned immunity or the immune system on medical immunology, so this part we won't go to the detail, but we'll talk a little bit about it. And today we'll focus on bacterial infection. And actually, this is just the major part of this topic. But in recent several years, with the discovery of more science facts, and we are not only studying infection, and actually we are not only studying pathogens, we are also studying other microorganisms. So that's what we'll talk about today. And on our first class, we mentioned that if you put all the bacteria in this pyramid, you fill this pyramid with all the bacteria, all the known bacteria. And at the bottom, that's the major part of all the bacteria. They are not pathogenic. For most of them, at the bottom, they are not pathogenic. So this part, they are not pathogenic. And those, we can say that they are normal microflora or microbiota, normal microbiota. They don't cause disease. Even they can live with us. They live on our body or they live inside our body. They don't cause disease. So these are not pathogenic. This is the majority part of them. And for some of them, some of the microbiota, usually they don't cause disease. But at very special conditions, they can cause infection. This we call them, they are potentially pathogenic. And this infection they caused are named as opportunistic infection. So this is part of them. Only very few of them on the top of the pyramid, very few number, they are always pathogenic. So these are called pathogens. So these are the major concern of medical microbiology previously. If you can find some old textbook, and in the old textbook, they mainly focus on this part, studying the behavior of pathogens, how they cause disease in human. So that's the old science. Now, yeah, they are still important. We need to study them because they are pathogens. But in recent years, we are focusing more and more on the lower parts. For example, the normal flora or normal microbiota, even though they don't cause disease, they are important. They can help human to stay healthy. They can even communicate with the nerve system, and they can communicate with our brain. They can regulate the function of our brain. And in the middle, this is opportunistic infection. Usually they are okay, they don't cause infection. But once they cause infection, the condition might be critical. So So now, in the subjects of medical microbiology, we are expanding our material much more than previous. And we are not only focusing on the pathogens, we are also studying the microbiota and the one in the middle, the opportunistic pathogens. Okay. So first we learn the majority part of this project, the normal microbial flora. This normal microbial flora are the population of microorganisms that inhabit the skin and mucous membranes of a healthy normal person. Here you should remember several points. First, it's on the healthy normal persons. See, this person must be healthy, a normal person, not someone can inhabit there. They are not guests there. They are living there. So they inhabit there. And it's a population of microorganisms, which means that it's not a single species. It's a composition. It's a large group of microorganisms. So this is the definition of normal flora. And there are two different types of a person who in the age group of 50 is different from that in a 2-year-old kid. So this is a resident microbiota. So the property of this group is that it's fixed types. So they are stable. This microflora is stable at this tissue, at this age. Okay, and the next type of microbiota is transient. Transient means temporary. It consists of non-pathogenic or potentially pathogenic microorganisms that inhabit the skin or mucous membrane for short times, maybe hours, maybe days, maybe weeks. So this is compared to the resident. These are short period of time. It is derived from the environment and does not produce disease and does not establish itself permanently on the surface. So it's only for a short time. For example, when you eat something, it's some raw food, and it can carry in some pathogen, some microorganism. And those microorganisms might be not pathogenic or potentially pathogenic. They can live in our digestive tract for a short time. This we call them transient microbiota. And I know for some of you, in your hometown country, you make different types of yogurt, and you know that in yogurt it contains a lot of bacteria, different types of microorganisms, and when you eat the yogurt, they can live in the digestive tract for some time, but they cannot live there for forever, for a long time. This we call them the transient microbiota. Okay, and generally you can roughly divide the balance is interrupted, is break. Some of the bacteria is gone. We say that the good bacteria is gone, and another group of bacteria will rise up, will dominate the area, and they can cause disease. We say that they are bad bacteria. They are bad flora, bad bacterial flora. But in our normal flora, they are there. They are living together, and maybe they are beneficial to other bacteria. So it's not perfect to call them good or bad. And this table contains normal bacterial microbiota on different tissues and different organs, like normal microbiota on skin, or the normal microbiota in the nose, or in the gastrointestinal tract, the digestive tract, or in the genital tract. So you don't have to remember this bacteria because it's not possible for you guys. But in the future, you might work on some of them, and you might focus on one of them to develop your career as a researcher. That's possible. So on different tissue, on different organs, the composition of the normal microbiota are different. Okay. So you want to know what's the role of normal flora. So what's the function of normal flora? Do they keep us healthy, or do they provide some nutrients? So that's what we are talking here. The first role of normal flora is that they can produce essential nutrients. Yeah, they do produce nutrients, including vitamin K, vitamin B. Some members of the resident flora in the intestinal tract can synthesize and aid in the absorption of nutrients. And we mentioned that in the digestive tract, the normal flora, the microorganisms, can help us to degrade the food we eat, and that can help us to get nutrients. And some of them can produce small molecules like vitamins, and those we can provide us essential nutrient factors. A second function is that some members of the resident flora on the mucous membranes and skin may prevent the colonization by pathogens and possible disease through bacterial interference. For example, if they share the same ligand for the receptors on the host cell, and the normal flora can take the receptors and bind the receptors so that the pathogen cannot use the same receptor to invade our host cell, or that they compete for nutrients. You know, the microflora and pathogens, they are all microorganisms. They might share the same nutrient. For example, iron. Iron in our body is kind of limited, so they need iron to survive. But if the normal flora takes the majority part of the iron nutrient, pathogens cannot grow up because it's limited. So this is a competition for nutrients. Or that the flora can contribute the mutual inhibition by metabolic or toxic products. That's the flora can produce some molecules which are toxic to pathogens, or that they can produce antibiotics or bactericin, like what we learned, colicin. And these antibiotics or bactericin can kill other pathogens or inhibit the growth of other pathogens. So this is the second role of normal flora. Okay. Another one is that the normal flora can stimulate the development of certain tissues, including like the lymphatic tissues and also the digestive tract. You know, the development of human including the development of certain tissues and organs, need the signal from the microorganisms, because those stimulation can help the tissue to grow, to differentiate, and to function. And without the normal flora, without the microorganisms, the tissue or the organ will be weak or thin. And this is observed in mice, and for sure. So if you live in a condition without any microorganisms, you cannot survive, or you are living with a very poor condition. The role of normal flora. Another one is that the normal flora can stimulate the production of cross-reactive antibodies. The normal flora and the pathogen might share some antigens, or the epitopes, so that the normal flora, even though they don't cause disease, they can still stimulate the immune system to produce antibodies. And those antibodies can recognize the normal flora, but they can also recognize the same epitopes on pathogens, so that these antibodies might neutralize the pathogens or help the immune system to clear the pathogen. So this we call cross-reactive antibodies. Okay, before we go to the next topic, I'd like to share with you a short video on microbiota, and this is a cartoon, and it will give us more clear idea, clear impression, and also include something we didn't cover. Okay, more about microbiota.
Information highway of our nervous system. Other examples are bacteria that stimulate immune cells in the gut, so they send a kind of alarm signal to the brain. Here it activates immune cells that help the brain recover from injuries. Since the brain decides when we eat, the microbiome is interested in a healthy brain. A new field of science is opening up here, and we're just on the verge of understanding how these complex systems inside our bodies interact. But we are starting to see how much our microbiome actually influences us and our behavior. Take depression, for example. Healthy rats fed microbes from the guts of depressed people began showing anxiety-like behavior and symptoms that look like depression. And in early 2017, a study linked the microbiome to intelligence by connecting a certain setup of bacteria in newborns with better motor and language skills. But it might also influence our daily lives. Tests with fruit flies showed that their microbiome influenced what kinds of food they craved. This could mean your microbes are able to tell your brain which food it should get them, although this is not a one-way street. The seed for our microbiome comes from our mother, but how it develops and changes is determined by what we eat. The organisms in our gut feed on different things. Some like fibers and leafy greens, others go for sugars and starches, and some love greasy fries and butter. Our gut is like a garden in which we constantly decide what will grow and blossom. If we eat healthily, we breed bacteria that like healthy food. If we eat a lot of fast food, then we breed fast food-loving bacteria. Life is hard, so we can get trapped in a vicious circle. You have a stressful time and eat lots of burgers and fries and pizza. This is awesome for fast food bacteria. They multiply and take up space from vegetable-loving bacteria. But even worse, they send signals to the brain to continue what it's doing. This makes you want more fast food, which breeds more fast food bacteria, which makes you crave fast food, and so on. This kind of self-reinforcing cycle could play a huge role in obesity. But it's important to stress that you can fight this process and reverse it by eating healthily and breeding more good bacteria. Beyond weight gain, our microbiome has also been linked to other serious diseases like autism, schizophrenia, and cancer. One of the earliest symptoms of Parkinson's is actually gut problems. If your body is overrun with bacteria that harm you, there is often only one solution. You bring in an army of good guys. That's very easy. You just transplant some healthy poop. You do that by literally transferring poop from a healthy person into your gut. This method is already used to cure diarrhea that's caused when C. difficile bacteria take over a gut microbiome. How our microbes make us healthy or sick. But whether we like it or not, we need our microbiome and it needs us. We'll never have our bodies to ourselves, but we have gained a powerful ally, if we can just keep the peace.
Yeah, it's an interesting video, and no wonder the video is good, and because it's supported by Bill Gates Foundation, and there are a lot of scientists working behind the video, and they show us the recent advances on the research in this field, and it displayed the results in such a easy way so that we can understand. Okay, so this is this microbiota. I think we still have time. We can talk a little bit more. Another type of infection is the opportunistic infections. Remember that pyramid. That's a group of bacteria in the middle of the pyramid. And this is the infection caused by the normal flora in the host environment. So those microorganisms are actually part of the flora. They don't cause disease usually, but at certain conditions. For example, if your immune system is weak or compromised, or that you are stressed and the immune system cannot function well, they can cause problems. For example, the host defenses are impaired or compromised. Like if the host is having a disease, or that the host is using immunosuppressive therapy. Sometimes when it's necessary, the doctor will give the patient some medicine to suppress the immune system, so that the immune system won't react too much. Or that the patient is receiving radiation therapy. The radiation therapy might destroy the healthy immune system. Or the patient is receiving chemotherapy, which is also influencing the immune system. Or, for example, the patient is having HIV infection, and the HIV virus can destroy the CD4 T cells, so that the immune system cannot function fully. Or it also can happen by the change of location of normal microbiota. When we learn the normal microbiota, we are talking about the certain age and certain area. If they are at this area when the host is at this age, it's normal microbiota. But if they change location, for example, if E. coli moved from the gut to our eyes, they can still cause infection. And if the bacteria from the gut moves to like our skin or other mucous membrane, they can cause infection. So this is a change of location of normal microbiota. Another possibility is dysbacteriosis, or the floral disequilibrium, or the balance is interrupted, the balance is broken. On the normal microbiota, it's a large population. It contains different species, and they form a network, form a stable state. And if this network is disrupted, some of them are killed, some of them are gone, then another part of them will rise, which can cause problems. And this usually happens when people use antibiotics or that they are hospitalized to receive treatment. Here is an example. So the colon is colonized by large numbers of commensal bacteria. So these commensal bacteria actually are the normal flora. This is the human gut lumen. There are a lot of bacteria or microbial flora or microbiota, and this is the human epithelial cell, and those bacteria line up in the lumen and can form a thick layer. It's like a micrometers, 50 micrometers or to 100 micrometers thick, and this layer can protect the colonization of pathogens. They are helping and keeping the lumen healthy. So this is the normal state. But if this patient is sick or for some reason, like maybe he has some trauma or wound and got some infection on the body, on the other part. So it's necessary to use antibiotics. So the doctor will give the patient some antibiotics to kill, uh, to inhibit the infection on the body. But the antibiotics is not selective. It can also kill these commensal bacteria, these normal flora. So it's disrupted, and a lot of them are gone. Now the pathogen or part of the normal flora is short of a C. difficile. This species will rise up, will dominate the gut, and they can cause infection. They can cause mucosal injury. So this mucous membrane is disrupted. It can cause injury. And once the injury happens, your immune system can sense the injury, so that it will send the immune troops, the immune cells, to the infection site. This includes the white blood cells like neutrophils, this one, the neutrophils, to the site to fight against the infection. And also this will damage the epithelial cells, so that the cells from the blood, like including the red blood cells, will leak into the lumen, and this can cause serious problems. So this is the problem caused by application of antibiotics. You want to use antibiotics to fight against infection, but actually administration of antibiotics caused infection. So that is once it was a serious problem in clinical practice, but now, as also shown in that video, you can use fecal transplantation to deal with this problem. And we know that it's because the normal flora in the gut, in the colon, is disrupted or broken. So you want to give this patient some normal flora, give them the microflora. See, so you can isolate the normal flora from a healthy person and transplant those microorganisms to the patient, so that the normal flora can set up, and then this pathogen will be get rid of. So this is the one interesting story we want to share here. Okay, before we take a break, we need to talk a little bit about the host defenses. Actually, there are multiple layers of host defenses. You know, the major part is immune system, what we learned on our immunology class. We won't talk about that in detail. But also, we have other layers. including our skin. Our skin is very tight, and the epithelial cells on our skin are linked with tight junctions. So that can seal us and isolate our body from the environment and preventing microorganisms, including pathogens, from invading our body. But except for the skins, we also have other mechanisms, like in the gut. Our gut is moving all the time. This is we call it peristalsis. Sorry, I cannot pronounce this word today. Or defecation. Defecation means that you get rid of the fecal from the lumen, and this movement can get rid of the um, pathogens or some pathogens in the lumen, in the gut, or in our respiratory tract. Respiratory tract, like in our lungs, we have the ciliaries, and the ciliaries are always moving. They are waving, so that they are waving in the same direction, so that they can push the waste or particles in the tract, outside the tract. This is a ciliary action. Or that we can cough, we can sneeze, and this cough and sneeze with the help of the air, so that we can take the waste out of the tract, and that can get rid of the pathogen in it. And also in the urinary tract, the urination, the urine can flush out the pathogen sticking on the tract. So these physical actions, physical procedure can help us to get rid of pathogens which can possibly cause infection. So this is different mechanisms of host defenses. Okay, we can have a break. Then we'll after break we'll talk about the major part of this lecture, the bacterial pathogenesis or bacterial virulence factors. Um, that will take a long time. So we'll be back in 10 minutes. If you have questions or have concerns or you want to discuss something, you can leave a message in the group and we can talk about it later. Okay, we'll be back soon. Talking about bacterial infection, we need to talk about several terms. We need to remember the definition of these terms, which are related to bacterial infection or bacterial pathogenesis. The first one is called pathogenicity. Pathogenicity is the ability of an infectious agent to cause disease. And we can say that one microorganism, one bacteria can cause disease. We can say that it's able to cause disease. We call this microorganism pathogen. While the other one, which cannot cause disease, we say that that one is not a pathogen. So this is the pathogenicity. Then how, on what level, on what grade they can cause disease, whether the disease is severe or not, we can use another word, virulence. Virulence is the quantitative ability of an agent to cause disease. And some pathogen might cause mild disease, while another one might cause a severe disease. This is a quantitative ability of an infectious agent. Okay, so these are the two terms related to pathogen and virulence. Okay, then when we talk about bacterial infection, we'll talk about different steps. The first step is that the bacteria needs to stick to the host cell surface. So they need to reside on the host cell surface. This we call it stick, or we call it adherence. So adhere to the host cell, adherence. Or some people call it adhesion. Or some people say this is attachment, a similar meaning, same meaning. This is the first step. They need to get close contact with the host cells. Then for some of the pathogens, they are okay to live just on the top of the cell. That's the infection. But for some other pathogens, they need to further invade into the host cell. Invasion to the host cell, inside the host cell, can benefit to the bacteria. First of all, if they are intracellular bacteria, they are hiding from the immune system, so that immune cells and molecules cannot attack this intracellular bacteria directly. And so it's a safer place. It's a heaven in the cell. And this process we call invasion. Invasion of bacteria or parasites or fungi or virus into the host cell. This is invasion. Another term we need to keep in mind is toxigenicity. This is related to toxin. The first toxin means toxin. The ability of a microorganism to produce a toxin that contributes to the development of disease. This we call toxigenicity. Some bacteria can produce toxin, and those toxin is involved in the development of disease. So this we call toxigenicity. Okay. Now we are talking about the bacterial virulence factors. They are infecting the host cell, the host body, in different steps, and at different steps there are different factors involved that enables them to cause disease, enable them to be virulent. We'll talk about the infection procedure from adherence to invasion to the molecules they produced, and some of them evolved the ability to inhibit the phagocytosis. So these are the anti-phagocytic factors. Or some of them can form biofilm even in the host body. And these bacterial biofilms can also cause further infection. Okay, now we are talking about those factors one by one. The first one is adherence factors. We mentioned just now that to set up an infection, the microorganisms need to get close contact with the host cell. So this we call adherence. This close contact is through either surface hydrophobic force. So the bacterial surface and host cell surface both are hydrophobic, and which can push them together. Or that the bacterial cell surface will have some charge, either positive charge or negative charge, which can help them to bind to the host cell. This is one type of adherence through hydrophobic force or the surface charge. Another type is through the binding of receptor and ligand. On our host cell, we have some receptors. Usually they are protein molecules or maybe other molecules. And on the bacterium side, on their surface, they can express some molecules. And this molecule we call ligand, or sometimes we can call them adhesin. This adhesin can bind to the receptor. This can help the bacterium to adhere to the surface. So this is adherence. Like E. coli, what we mentioned previously, they produce type 1 pili. This type 1 pili is the common pili, which can bind to the host cell surface, mannose as a receptor. They can bind to it. or the Streptococcus pyogenes. This is the coccus bacteria, Streptococcus. They produce a protein called F protein, which contains a lipoteichoic acid. This lipoteichoic acid can bind to fibronectin, a human protein produced on the surface. So this is a receptor and ligand binding, which can help the pathogen to adhere to the surface. And this picture, this cartoon, shows us the role of pili in adherence. Like this bacteria, they don't express pili. They don't express pili. So this is in the urinary tract. So they just stop on the cell surface. When the urine flush comes, it easily takes the bacteria away, flush the bacteria away. See? So this is the bacteria without pili. But if the bacteria can express pili, this pili can help the bacteria to adhere to the host cell surface with a higher affinity. Adhere tightly. And again, when the urine flush comes, it cannot take the bacteria away. It cannot flush them away. So this is the role of pili for adherence. So make them more resistant to liquid flow. So the next step is penetration and spread. For some pathogen, just adherence to the surface is not enough to set up infection. They need to further get into the cell. They need to further get inside of the cell. This step we call penetration. And once they are inside the cell, they can multiply. They can replicate. And they need to, the nutrients in one cell is also limited. When they get higher numbers in the cell, they need to infect adjacent neighboring cell. So they need to spread from one cell to another. This step we call spread. So this is another the next steps. And this is the famous picture of Listeria monocytogenes. Listeria is a gram-positive bacteria pathogen, which can cause food poisoning. So first they adhere to the surface through different molecules, and then they invade into the cell. Once they are inside the cell, they can take advantage of actin polymerization. We know that when you learn the cellular biology, cell biology, we know that actin forms the cytoskeleton of our cell. And the actin filament is a dynamic filament. It grows from one end and degrades from the other end. And the bacteria can hire the actin filament at its tail site. So this is a filament of actin binding to the bacteria. And this tail is not stable. It degrades and grows from one end and degrades at the other end, which kind of like a chain which can drive the bacteria to move, which can push the bacteria to move, so that it can push the bacteria to move inside the cell and also push the bacteria to move through the membrane and spread into another bacteria. So this is a very famous picture in medical microbiology. This is not just drawn. The scientists actually saw this happening in the cell with electron microscope. So this is what was published in the paper. In the middle is a model. It's drawn a model, but the outside images are just the real image under electron microscope. And once the listeria is inside the cell, and first they hide the actin around them. They form a coat of actin surrounding them, and then gradually the actin can form a tail behind the bacteria. Here, then the tail can grow longer and push the bacteria to move, like this one. The actin tail behind the bacteria can push the bacteria to escape from the infected cell. And sometimes these are the two cells. It can push the bacteria from one cell and then invade to another one. So this is spreading. Okay. The next topic we'll talk about toxins. Pathogens, different microorganisms, they can produce different types of toxins, and according to the material property of the toxins, there are two types of toxins. One is called exotoxins, the other one is endotoxins. For endotoxins, we actually learned a little bit when we learned the bacterial cell structure and components. And we know that endotoxins are actually the lipopolysaccharide, short for LPS. And we learned the structure of LPS and the components on the molecule. For exotoxin, they are proteins. We'll talk about exotoxin first. So exotoxins can be produced by either gram-positive and gram-negative bacteria. Most exotoxins are protein molecules, so they are proteins. Usually they are enzymes. And most exotoxins are heat sensitive. Here, most means, usually they are heat sensitive, but for some exotoxins, they are heat resistant. You generally simply increase the temperature doesn't kill the protein molecule, doesn't inactivate the protein molecule. The action of the exotoxin does not necessarily require the presence of the host bacteria in the bacteria in the host. So even though through cooking or some other procedure you can kill the bacteria, you can inactivate the bacteria cell. Once the bacteria release exotoxins into the environment, they can still cause disease. So it's independent to the live bacteria. Like some time ago, there was a news in China. A family got food poisoning, and I think four out of 6 people in that family were killed. And that was because of some exotoxin, because the food was not fresh, and some type of bacteria grow in that food and released exotoxin. Even though they cooked the food thoroughly and killed the bacteria, but the toxin in it still can kill people. And exotoxins are usually high toxicity, with high toxicity. And this toxic activity usually is tissue or cell specific. They are tissue or cell specific because these toxins need to recognize a receptor on the cell. Without a receptor, they cannot invade into the cell. And because they are protein molecules, and we know that protein molecules are good antigens, so they are strong antigenic. So they are strong antigens which can stimulate the host to produce antitoxin. And now we call this antitoxin. Actually, they are antibodies which can neutralize toxin. Antitoxin is the first name for antibodies, because at that time, that's over 100 years ago, at that time people know nothing about antibodies, but they realized that those material or those substance can neutralize toxin, so they call it antitoxin. Here is how the exotoxins invade into the host cell. Usually, for most of the exotoxins, they have the similar structure, which is called AB toxin or AB structure, which means that there are two subunits, A subunit and B subunit. The A subunit is the toxic subunit, which can carry out its effect, while the B subunit is a receptor binding ligand, which can mediate the adherence of the toxin complex to a host cell and help the toxin to get into the host cell. Like in this cartoon, this one of the AB toxin. There are two subunits. Let's see from the beginning. Two subunits. The big one is A, the small one is B. B can bind to the receptor for B component. Bind to a receptor and then internalized into the cell, and then A subunit is released. Then the A subunit can carry out its toxic activity. So this is the exotoxin. Exotoxins, they are proteins we mentioned, and we human can actually manipulate those proteins, because we learned that they are strong pathogenic, they are strong antigenic, which means that we can manipulate the molecule and make them as antigens. But we need to get rid of the toxic activity. We can treat an exotoxin, and so that we can remove the toxic activity, but still we can maintain the antigenic activity, so that we can use that modified molecule or manipulated molecule as a vaccine. And this type of vaccine we call it toxoid. It's generated from exotoxins, but denatured by the chemical formaldehyde. You know, the protein is denatured, it's not functioning anymore, it's not toxic anymore. It lacks toxic activity, but still induce protective immunity, and we can use it as a vaccine to stimulate the immune system to produce antibodies, to produce memory cells. So this is one type of vaccines we are still using in clinical practice. And I guess that when you guys are young, are kids, you received some vaccines in the form of toxoid, like the vaccine against diphtheria, which is a toxoid. Okay, so this is exotoxins. The exotoxins can be classified into different groups. The first group is neurotoxins. These neurotoxins interfere with proper synaptic transmission in neurons, so that it can block or inhibit the neuron activity, working on the neuron cells. Another toxin is called cytotoxin. Cytotoxin, you know, cyto means cell. So cytotoxins can inhibit specific cellular activities, such as protein synthesis. So this is cytotoxin working on the cell. Like diphtheria toxin is one type of the cytotoxin. Another type of toxin is called enterotoxin, and we know the word entero means the digestive tract, the intestine. So this type of toxin interfere with water reabsorption in the large intestine. irritate the lining of the gastrointestinal tract, which can cause like diarrhea or other symptoms in the digestive tract. The different classes of exotoxins. Okay, a little bit about endotoxin. We learned this previously twice, I think. They are a component of gram-negative cell wall. So keep in mind that endotoxin only exists in gram-negative cells. They are on the outer membrane of gram-negative cells. Its name is lipopolysaccharide. It contains polysaccharides and also contains lipid A. It's not proteins, it's not enzymes. The endotoxin may be released from the cell wall as the cell dies. So usually when the cell is alive, endotoxin is the component structure on the cell wall. That is why we call it endotoxin, because it's on the cell wall. But when the cell dies, it will be released. And once it's released, it can carry out its function or can affect the host cell, host body. Endotoxin is heat stable, which means that simply increasing the temperature doesn't kill the LPS molecules. If you want to kill the LPS molecules, you need a high temperature, maybe like around 200 degree, 170 degree, and bake it in the oven for several hours. That's what we do in the lab to inhibit, to inactivate LPS. They are not cell or tissue specific because they don't have to be recognized by a specific receptor. Does not stimulate the production of specific antibodies in the host. And also, endotoxin from different bacteria carry out a similar function, similar effects, because they share the similar structure. This is endotoxin. Endotoxin effects include fever and also like leukocytosis, hypotension and shock, complement cascade, DIC, and also other problems. The procedure for the endotoxin to affect human cells is shown in this picture. We can see that this is a 1 gram-negative bacteria. And when this bacterium die, it will get lysed. And the lysis of this cell will release LPS molecules. LPS molecules first bind to the LPS binding protein. This is the human protein. And this protein will carry the LPS molecule to the host cell and recognized by CD14 and Toll-like receptor 4. And once recognized, it can stimulate the host cell, usually it's a macrophage, stimulate the macrophage to express cytokines. These cytokines include L1, L6, L8, TNF-alpha. Those cytokines are pro-inflammatory cytokines, which can promote inflammation. And this will lead to further effects, including those symptoms, either fever, DIC, and also activation of the complement system. So it will cause a lot of problems. So this is the endotoxin effects. This form or this table compares endotoxin and exotoxin in different properties. Their chemical nature, relationship to cell, denatured by boiling or other properties. I'd like you to keep an eye on this form when you learn different types of toxins. This comparison between different toxins can help you to remember the properties of each toxin. Okay, so this is a brief summary of what we mentioned just now. Here is a short video, and we can watch this video. Generally, it's a brief summary, and it covers what we talked about just now. Okay.
You have toxin circulating in the host. Now, if that's a human, this is going to be a very dangerous situation, especially if you're talking about things like neurotoxins. Diseases such as the Clostridiums, tetanus, botulism, have very potent exotoxins which can potentially kill. Now, over on the flip side, we have what are called the endotoxins. So the endotoxins are typically found in gram-negative part of that cell wall. Okay, so it's not an additional release of toxins that end up entering the body. Again, these are going to be gram-negative bacteria, so your gram-negatives will have endotoxins. Not all endotoxins are the strongest things out there, okay? But if they get into the blood or a place where they're not supposed to be, that can be a very dangerous situation. And right here what we have would be, what if you have a gram-negative? All right, you know that they're going to have endotoxins automatically. But they can also have exotoxins. So I represented them, the N's for the endotoxins, the X for the exotoxins. So this is a pretty powerful gram-negative. Okay. Over here, I had showed you a very powerful gram-positive because the gram-positive had the endospore as well as the exotoxin. Remember, only two genus that we're going to talk about are medically relevant with endospores. You have the Bacillus genus and the Clostridium genus. No other medically relevant genus out there with regards to that. So those are your exotoxins, your endotoxins, and then again, a gram-negative could potentially have both. A gram-positive is only going to be able to have an exotoxin. It cannot have an endotoxin because it does not have a lipopolysaccharide. So I hope that clarifies the toxins for you.
Okay, can you hear me? I guess so. Okay, so basically that's what we are covering today for this topic. And next time we'll start with enzymes which are produced by pathogens and which are involved in pathogenesis. And yeah, that's all for today. Thank you and have a nice day. Remember that we don't have a test this week, and so that you can enjoy your weekend. Okay, have a nice day.